Hello, I'm Jörg, and in this video you will learn more about LC-MS, what the technology does, how it works, and why it's so important in pharmaceutical development. From API development to finished drug product, liquid chromatography, mass spectrometry, or short LC-MS, plays an important role. It's a versatile and powerful analytical tool in pharmaceutical science. Today's drug developers need to understand every component of a drug product's formulation. Is it safe? How does it behave over time? What about API degradation products? LC-MS plays a central role in answering questions like these. So how does it work? It's actually a combination of two complementary techniques. First, liquid chromatography is used to separate the components of a complex solution containing the API, its excipients, and possible impurities. Chromatography separates these substances step by step as they pass through a column. Second, mass spectrometry is used to identify each separated component by measuring the mass-to-charge ratio of ionized molecules. Each molecule produces a characteristic signal like a molecular fingerprint. Scanning for these signals allows precise identification and quantification, even at very low concentrations. As you can tell, modern LC-MS systems are highly advanced. They can analyze simple small molecules, of course, but also complex biologics like oligonucleotides and proteins. The instrumentation and data processing used for LC-MS are both evolving rapidly, enabling us to characterize highly complex mixtures with great accuracy. So why is LC-MS so important in pharmaceutical science? First, unique identification of molecules. During R&D in drug substance manufacturing, it is essential to confirm that the intended compound has actually been produced. With LC-MS, molecular structures and sequences can be verified and component identity can be confirmed with great consistency. Second, detection of trace-level impurities. Purity, of course, is critical. LC-MS can detect extremely small amounts of unwanted substances, such as residual solvents, degradation products, or process-related impurities. This makes it essential for impurity profiling, stability studies, and demonstrating that manufacturing processes do not leave harmful residues. Third, understanding how drug products change over time. During production, storage, and distribution, products may degrade or change. LC-MS monitors these processes in detail. It identifies degradation processes and tracks molecular shifts. This helps demonstrate that the product remains stable not only during manufacturing, but throughout its entire shelf life. Fourth, supports safety in pharmacokinetics. To define accurate dosing, we must also measure how a drug behaves in the body. LC-MS can quantify very low concentrations in biological samples such as blood plasma or tissue. This precision is essential for pharmacokinetics and toxicological studies. Finally, of course, quality in manufacturing. Under GMP conditions, validated LC-MS methods are used extensively for quality control. The technology supports release testing, ongoing stability programs, life cycle management, and more, verifying identity, purity, and consistency across commercial batches. As you can see, LC-MS provides critical molecular insight throughout the drug product life cycle. And as drug products become more complex, it's a technology that will become more important in delivering safe, effective, and high-quality therapies to patients worldwide. That's it for today. Please leave a like if you enjoyed this video and subscribe to our channel for more updates. If you'd like to know more about the topic, check our website. See you next time.